Okay, so first I would like to thank the organizers for this kind invitation, and I will try to move to pick your attention to the engineering we are doing to try to evolve or to generate microbes, bacteria that could be used for some therapeutic applications. So our approach is, let's say, a modest one, trying to generate modules that can be combined in, let's say, a chassis that of your choice. And so it's not kind of a radical issue like complete engineering of the genome or changing the bases, etc. So we more focus on developing these particular modules. So I will concentrate today in our work that we have been doing to provide targeting adhesion properties to E. coli and also to provide the expression of injectisone to non-pathogenic E. coli strains. So as you probably know, the use of bacteria in medicine has been quite limited, and certainly most applications that you still see in the market comes from the use of probiotics, natural strains that have certain properties. So they are considered natural, so they from yogurts or from, let's say, certain isolates from a patient, etc. There are certain strains that are considered beneficial and are given to for certain pathologies or to protect enhance your immune system. And there are other major applications, which is basically the use of attenuated pathogens to induce immune responses and then produce vaccines, or to induce as vaccines or induce a vaccination. Since recombinant technology appears, there was clearly an interest in the modifying bacteria to produce molecules, not only in vitro, but also in vivo, maybe to enhance these properties that are sometimes found in beneficial bacteria. And in fact, if you review the literature, there's quite a few examples in which the people has been using traditional recombinant engineering to modify probiotic strains or to modify non-pathogens or even attenuated strains of pathogens to deliver certain molecules of interest, like, for instance, interleukins or antibodies against inflammatory diseases, some cytotoxic proteins against cancer, because bacteria are able to grow in solid tumor preferentially when they are administered systemically, as you will see later. And also other molecules that could intervene against infections, and viral or bacterial infections. So you could have a probiotic that could protect your mucosal surfaces against this type of infection, or even deliver certain molecules to cells in your gut, so you can reprogram the metabolisms or some molecules produced by the epithelial cells that are in contact with these microbes. And this is areas in which there's been work, there's many groups that are working on developing this type of applications. So, well, a consensus, let's say, for us, it would be to have, with this synthetic biology, you can generate modules of different types. And so ideally, synthetic biology can provide a way to combine all these modules in a perfect chassis, so you will not need to use a natural strain, which is partially beneficial, but will have also other properties that are unknown. Okay, so we could ideally think on introducing in a chassis that is maybe indicated for a certain application, all number of modules to sense the environment and response to that. Or, for instance, by delivering a protein, we can also introduce modules that would allow the bacteria to move toward certain signals, so targeting the bacteria to certain areas, or introduce reporters that could indicate us that the presence of a certain molecule or the presence of a certain disease. And all could be contained by certain mechanisms of containment. So you can introduce in your bacteria all these type of things. I would talk, as I mentioned previously, on a module that we have developed for targeting the specific addition of E. coli and to cells and surfaces, and also later about a delivery module based on Injectisomes. So why we focus first on this type of modules to target the addition of bacteria to certain cells? Because we consider that this is an essential property if you want to use bacteria for therapeutic applications, is to have the possibility to deliver your bacteria to the type of cells that you are interested in. Or maybe in some cases you will have a pathological disease and cell that is infected by a virus, so you have an antigen of a virus there. Or it can be a tumor cell and you will have certain protein expressed. But also, in addition to this type of application, you think that these modules could have other applications also in vitro, let's say on abiotic surfaces. So you can maybe develop this type of adhesins against surfaces so that you can help your bacteria to attach to a certain surface, for instance, in a biosensor. Sorry, I think it moves in the wrong direction. Yeah. Okay. So if you see what's in nature, and so what happens, how the bacteria adhere to the different surfaces, is they have developed a number usually of proteins or polymers of proteins that are assembled on the surface of the bacteria, and that allows the bacteria to recognize certain structures on the surface. Usually, as any strain, any natural strain carries multiple adhesins against different, with different specificities, and so it's difficult to reprogram a natural strain if you do not delete partially the major adhesins that it contains. Another problem with natural adhesins also is that the specificity it is kind of broad and low, and this is because it frequently recognizes carbohydrate, and as you have heard from the previous talk, carbohydrates are frequently found in many proteins, so bacteria will have the tendency to bind to more than one tissue, and so this are more than one cell types using natural adhesins. So you have to really engineer something new if you want to be more specific. And so we thought of just based on what is known about adhesins, as I told you, that can be anchored on the surface. We play with different possibilities, but we wanted to reduce this to, let's say, two modular parts. One is a domain that will carry the adhesion property, and the second will be a domain that anchors this to the surface of the bacteria. So what will be the ideal properties of this type of domains? Will be high affinity and specificity, of course, and additionally that it would be something that you can select from a library, okay? So you can later, just simply by changing this adhesion model, you will be able to target to different surfaces. So you will have a method of selection and a large library of this type of domains. So we thought of using, why not, immunoglobulin domain, because natural adhesins in bacteria frequently have Ig-like domains, like in Fimbria. This is a FIMH domain of a pili, of type 1 pili in E. coli, and it carries an Ig-like form. Okay, so it's possible to use a human or other type of antibody-like domains to replace for natural adhesins. And we decided to use, after testing different types, this type of antibody molecules that are derived from camel antibodies, which carry all this specificity in a single domain of the variable VH domain. These antibodies are found in camelids. They were discovered in Belgium by the group of Serge Mulderman years ago. And this has very interesting properties in binding and solubility, etc. And so there are, and one thing that attracted our attention also was that they are simple and you can express them very well in bacteria. Okay, and in terms of selection, this system also allows to easily select the specificity you want. I just simply summarize the standard methodology that is developed, but there are other alternatives. Just after immunization of an animal of this type, you generate a library of genes from the B cells, you clone them in E. coli, you can infect with bacteriophages, and make a pool of bacteriophage and select the specificity of the clone that is recognizing the antigen of your interest. This is a simple scheme for basic technology that it was developed years ago. So we decide to use this type of antibody-like molecules as adhesion domains, and we later, of course, co-developed a way to anchor these domains on the surface of E. coli, and we tested different anchor domains. I'm going to make the story very short. The domain or the proteins that end up to be the best ones were derived from pathogenic E. coli, enteromorrhagic E. coli strains that are called intimins. Intimins are proteins that are expressed on the surface of E. coli, of these strains, and allow the bacteria to attach to the surface of enterocytes. It carries a barrel, a domain that anchors in the outer membrane, and it displays Ig-like domains toward the surface. And this is shown here. This is the outer membrane of the bacteria. Here is the host cell. And these bacteria attach to the enterocytes using intimins, which display these Ig-like domains for contacting a specific receptor that the bacteria injects into the enterocytes. So it's a protein that has a certain specificity, a clear specificity for a specific protein that is not found normally in the mammalian cell. So we end up with this type of constructions with an outer membrane anchor domain and this antibody domain. And we started to express these modules with plasmids, standard plasmid that you can induce in a strain that lacks the major fimbria type in E. coli, so they do not attach to mannose residues. And we found that they were correctly expressed, displayed on the surface. We introduced tags for immune detection, and that they bind correctly the epitope on solution. And we also tested if they could somehow derive the addition of the bacteria to a surface coated by the antigen recognized by the antibody, in this case. So we coat ELISA plates with different antigens toward which we have nanobodies against them. And as you can see, if you incubate these surfaces with E. coli bacteria expressing these adhesins on the surface, you have bacteria attached to the surface of the plastic coated with a fibrinogen or GFP. If you have adhesion, these adhesins recognizing GFP or human fibrinogen on the surface. So we decided to move from the plasmid to the chromosome to avoid antibiotics and to avoid the complexity of making induction, etc. So what we did essentially was to recombine our plasmids into the chromosome, substitute the PTAC promoters by constitutive promoters, and delete simultaneously that we introduce other reporters like bioluminescence in our strain. We delete other adhesins that could interfere with the addition process. And one thing that we tested, and for us was very important, was to see at the same time that we introduced this construct, we remove all antibiotics. Okay, but one thing that we tested is that not only the expression of our construct was okay in the chromosome, but also the growth and the viability of the bacteria was perfect. So it was essentially growing at the same rate and it has the same viability than the strain that doesn't carry these constructs in the chromosome. And as you see, the expression of the construct is maintained throughout days of continuous culture, okay, without any requirement of any selection pressure. So it's just growing the strains in LB. Okay, so we wanted to test this with cells, and so since we had this type of certain antibodies, nanobodies against GFP and also against the translocated intimin receptor, which is an antigen of this EHEC, which is not found, as I told you before, in HeLa cells or in human cells, in any part of your body. So we thought this could be nice examples to test the addition of these strains to our cells. So we make stable transfectants of HeLa cells that express on the surface the antigens recognized by the adhesins. In this case, it was GFP, displayed on the surface, anchored by a transmembrane domain. And this is the region of this antigen that is TIR, recognized by this adhesin, and it was anchored to another fluorescent protein that is not doesn't cross-react with the nanowire against GFP. And we tested initially growing cells in vitro on plates, the untransfected cells, or cells that expresses GFP or TIR on the surface, and we incubate these wells with our strains, the one that is a control or the one that expresses the adhesins against GFP or TIR. And initially, as I told you before, we introduced this bioluminescence, so you can see all wells that are producing light. So, and once you wash, to just see what are the bacteria that remain on the wells after washing, you just see that only the bacteria that expresses the adhesin against GFP binds to HeLa cells expressing GFP on the surface, and only bacteria expressing this antibody against TIR binds to the well that carries HeLa cells with TIR on the surface. As you take a look at what is inside, you would be able to see it better. So you will have cells. This is a field in which you can see some untransfected cells and other cells that are expressing the GFP. And this is a stain for E. coli. And as you see, E. coli binds to cells that expresses the GFP on the surface. And the control strain that recognize a different antigen will not bind to these cells. Okay. And a nice control is these non-transfected cells that, as you see, are clean of bacteria. This is more or less the same, but with confocal microscopy, selecting a field in which you have a transfected and non-transfected cells, and you can clearly see the number of bacteria surrounding your target cell. And the same happens in the case of the other adhesin against TIR. You have a cell that is expressing TIR, it's fully covered with bacteria. So another important thing is the speed at how this happens. And we make a video to see how in a time frame of less than 3 minutes of infection, you have here a cell that is non-transfected, and the bacteria scan a little bit the surface and then escapes. And whereas in cells in which there are expression of the target, bacteria go and attaches to the surface and stop moving. And this is something that occurs, as you see, in less than 3 minutes. So it's very fast. We wanted to test this a little bit in vivo. And as you probably know, bacteria, especially anaerobic bacteria, has been used since more than a century ago to try to eliminate tumors, because certain bacteria, specifically anaerobic, can grow in anoxic areas in solid tumors. And this is work that was initiated more than 100 years ago, but as I said, there are groups like Neil Forbes from Michigan who are engineering salmonella strains and others to be able to deliver specifically molecules of interest in the tumor. One issue with this type of let's say, experimental approach against cancer is the dose that is safe to be administered. Of course, you need a bacteria that is not pathogenic, but also you need a low dose to avoid a systemic infection. But at the same time, the dose has to be high enough so that you guarantee the colonization of a tumor by the bacteria. So we decided to test whether the expression of these glues, this adhesin on the surface of the bacteria, helps the colonization of the tumor. And so we established with our cells a model, experimental model, based on HeLa cell expressing GFP. So you can, by subcutaneous injection in nude mice, you can generate a solid tumor in the flank of the mice. And later you can introduce your bacteria by systemic infection in the tail vein of the animal. And it is reported that in this type of approaches with bacteria colonizing tumors, 3 to 5 days after injection, there's a peak of the proliferation of bacteria in the tumors, whereas other organs usually have less number of bacteria, as you will see. And so we decided to test whether our bacteria were able to colonize these tumors with expressing this GFP, and it would be a better targeting of our strains. And initially we just simply use the dose that is reported by the people that is using Salmonella or even E. coli K-12 to access a tumor, a solid tumor that is in the range of 10 to the 7. And as you see, both are strain that is targeting the tumor that expresses GFP and the strain that is a control. Colonize it equally well. 6 out of 6 animals were, the tumors of these animals were colonized efficiently and with having a bacterial proliferation toward 10 to the power of 8 per gram of tissue of tumor, whereas other organs like liver and spleen were almost clean of bacteria. Okay? So that means that at a high dose there's no difference. So we wanted to test what happened if we reduce the dose, okay? If we really force a condition that is not optimal for the bacteria to colonize those tumors. And so we reduced the dose to 1% of standard dose using 10 to the 5. And as you see from this slide, now then there's a significant difference between the strain that carries an adhesin that targets the tumor, in which in these animals 8 out of 9 animals were colonized, the tumors of these animals were colonized by the bacteria efficiently, whereas in the controls only 2 out of 9 or 3 out of 9 using the wild-type strain that carries non-specific adhesins, natural strain, were colonized. So this engineered strain significantly enhanced the colonization at lower doses. So you are able to target more efficiently bacteria using a lower dose to a tumor. And this is a control of a specificity. We introduced a group in which the tumor were generated by HeLa cells that were not expressing GFP, and in this case our strain was not able to colonize this tumor more efficiently. It was like in the control, identical to the control. So to conclude a little bit this part of the talk, I show you these constructs, these adhesins that can be expressed constitutively from the chromosome of the bacteria, and that can allow you to target specific cells or surfaces or tumors in vivo. Okay? And we believe this, we are developing adhesins that will target real tumors with specific markers and that in areas in which bacteria may access and may deliver specific molecules toward the tumor cell or in the tumor environment. So I then will focus on one of these delivery machineries. And as you probably know, bacteria had evolved a number of protein secretion systems, especially gram-negative bacteria, to translocate proteins across the inner and outer membrane. And one of these, some of them are simple, but some others are extraordinarily complex and allow direct translocation from the bacteria to another cell. And this another cell, for this particular type of protein delivery systems, usually is a eukaryotic cell or a mammalian cell, plant cell. And so many pathogens from especially gram-negative strains from Pseudomonas or from E. coli, Salmonella, carries complex machineries that are called Injectisomes for the type 3 secretion system that allows the translocation of specific proteins toward the cytosol of the infected cell. And these effector protein that are translocated by this machinery can make multiple functions in the host cell, but helps the infection to proceed. They can target cell cycle, they can target inflammation, they can target the cytoskeleton and mitochondria, so they have multiple possibilities to target. Okay? And usually these machineries are coupled to dedicated ATPase that energizes the system for translocation. They carry also, the effectors that are translocated carries a short N-terminal sequence that is not cleaved, but is recognized by the machinery to be translocated toward the cytosol of a mammalian cell. So in a way, this is like a syringe, a molecular syringe for translocation of proteins, and there's clearly an interest in using this type of devices toward a specific delivery of molecules in the cytosol of mammalian cells. So we decided to try to assemble this type of injectisome based on the injectisome that are expressed by enteropathogenic E. coli strains, which, as I mentioned previously, help the bacteria, these pathogenic strains, to attach to the enterocytes. So they are syringe-like complexes, as I showed you before, but the significant difference of this type of injectisome is that it extends a filament, a very long filament that can be up to almost a micron long, to access the enterocyte even from a distance. So he can inject proteins not even with a close contact. He can inject protein initially from a distance, and then it introduces, for instance, this translocated intimin receptor, and then it binds intimately to the enterocyte later. Okay? So these filaments have a channel in which the proteins are supposed to be translocated through this pore of this molecule. So this is a scheme of how EPEC attaches, injects the translocated intimin receptor, and form this attaching and effacing lesion at this particular interaction, which in this particular case, the tear is also able to signal and to reorganize actin on the surface of the enterocyte, and so the bacteria will be let's say, sit on a pedestal of actin. So it's something that is very easily seen, is that the bacteria remains extracellular, but the actin polymerizes just underneath the bacteria, having like a pedestal-like structure. Bacteria not only introduce tear, it introduces, as I mentioned previously, many other effectors that has other functions, this type of bacteria. So initially our interest was with antibodies, but because, as you know, antibody molecules are usually targeting extracellular receptor or soluble antigens, just simply because they are accessible. But there are many other targets inside the cell which are important and which are difficult to access using an antibody strategy. So we decided to, so we thought that since E. coli can be used for selection of antibody molecules, why not use the same strain also for delivery? But as I said, this type of injectisome could deliver many other proteins of interest, not only the effectors. So we did some work with the pathogenic strain just to test that the syringes were capable of translocating small antibody fragments like the nanobodies that I mentioned previously. And as you see, we tag the nanobodies with a sequence that is recognized from a natural effector, and we could see the secretion, in this case, toward the medium, dependent on the type 3 system of the antibodies fragments. And we made different experiments to show that they were active, they were binding the antigen with similar affinity. And these are just simply mutant in which you knock out the ATPase, and then without this ATPase activity, you don't see any secretion of the molecule or other proteins that are forming this filament and the pore. And we also test that these antibody molecules could be, with this system, could be injected into mammalian cells, not only secreted with the system. We did different experiments. Maybe the more visual is this one, in which you use a reporter that is an enzyme, beta-lactamase, that can degrade a substrate, a fluorescent substrate in the infected cells. So this, what you see here, are infected HeLa cells that are, you don't see the bacteria, you just simply see the HeLa cells. And if the beta-lactamase is inside the cytosol of the cell, then this substrate that is green and of the beta-lactamase turns blue because of the enzymatic activity of the beta-lactamase. And so we use this assay to test whether we could translocate also the nanobodies, and we test several of them, and we also see translocation of fusion between the nanobodies and the enzyme inside the HeLa cell. So it was clear that there was a potential to translocate heterologous proteins using the injectisomes of EPEC. But the problem was that EPEC is a pathogen, so you are not going to use a pathogen at all for introducing anything, especially because, as I told you, EPEC not only introduces the protein that you are interested in, it introduces over 26 effectors, other proteins that you are not interested on. So we decided that the best way to go would be to move the injectisomes to a non-pathogenic strain and see if we could assemble these complexes, the complex delivery, into a K-12 strain, generating something that we call as synthetic injector E. coli strain, okay, CEC. Just to continue with this EPEC, EHEC, CEC strain, the nomenclature that they saw. So how we introduce this into K-12 was, so how we envisioned the introduction was not easy. It was previously reported that you can clone the whole island into a cosmid, and then you could see some assembly of the type 3, but it was very weak, and partially because the regulation is not maintained, and so the loci that encodes all structural proteins of this injectisome is located in a region of about 35 kilobases that is called the locus of enterocyte effacement, and it carries the structural genes required for the assembly of this machinery, but it also carries other genes that might not be, they are not interested for synthetic approach, like regulators or the some effectors that are being injected and that you don't want to have there. So we decided to pick from these loci only the genes that were required for translocation. And since we wanted to have a good expression and a controllable expression, we decided also to remove the promoters from our constructs. And so what we decided was to remove the regulators and remove the effectors and promoter regions. And so we just decided to amplify the genes that were coding essential genes to assemble the injectisome in a functional way. So we amplify these engineered operons, okay, without any, as you say, only the open reading frames, with no promoters, no regulators, and no effectors. And we introduce these frames of fragments into vectors that carry PTAC promoter to induce the system by addition of IPTG initially. And as we are not, we don't want to work with plasmid or antibiotic resistance, as I mentioned previously, so we decided to integrate all our constructs in the chromosome at different positions. And in fact, what we did was to integrate them in replacing natural adhesins found in bacteria. So our strain was further deleted of natural adhesion factor and during the process of integration. So we integrate these 5 operons in the chromosome, replacing these cryptic adhesins found in K-12, and we indeed generate a different strain, an additional strain that lacks the promoter in the first operon of the system, so that we could have a control of the expression of a control of the specificity of the translocation of any protein that we wanted. So this is an experiment that in which you can see growing the strain and comparing with the production of these injectisomes in EPEC. You compare it with the production in the engineered strain with addition of IPDG. And these are proteins, these ESPD, B, and A are proteins of the filament that are in fact translocated through the injectisome and form this filament to help to recognize the mammalian cell and then form a pore for protein translocation. So once you see this protein, is that the type 3 system is active, and as you see, the 3 proteins B, D, and A are also found specifically when we induce in the supernatants of our engineered strain, and you can recognize them by specific antibodies. So we detect by Western blot different components, but we decided to go a step further and try to purify the injectisomes of our engineered strain and compare with what we could purify from enteropathogenic strain. And as you see, we could detect filamentous structures of different lengths that are compatible with the antibody and that are recognized by antibodies against this injectisome from EPEC and from our engineered strain, which are very similar they look very similar. And so our strain is probably assembling bona fide injectisome of EPEC. So we tested by different methods how can we, if our strain is able to translocate proteins into mammalian cells, acting really as a syringe. And since I previously mentioned the one that is more easy to test probably is the tear effector. Why? Because one is the first protein that is translocated naturally by the pathogen. It inserts in the membrane and then it polymerizes actin. So there's a simple readout to detect if there's been translocation of this protein by the type 3 is that the actin polymerizes underneath the bacteria. So we decided to test whether our strain was able to translocate tear. And for that, we introduced tear with a specific chaperone that is required for its secretion, and also intimin now on the surface of the bacteria using amplifying this fragment of the Li. So we will have a strain that, in addition to expressing all the components of the injectisome, will carries this from the chromosome, carries the effector protein and the intimin for specific recognition of tear. And this is a confocal image in which we compare the pathogen, the EPEC strain, in which, I don't know if you are able to see something, but the bacteria are stained in both cases with this cyan color. So it's what you see here are cells infected that almost it's difficult to see the actin because it's stained in red and it's hard to see. But then you have to believe me that the bacteria, in this case is EPEC, there's a small accumulation of actin underneath the bacteria. Okay, this is a strain that lacks the ATPase. So in this particular strain, you will see bacteria attached, but there's no actin accumulation. There's no red spots underneath. And these are the engineered strain in which you see the bacteria in cyan, and then all this red is the polymerization of actin underneath the bacteria. So meaning that our engineered strain was capable of translocating this protein and into the mammalian cell in a functional form. So it was able to signal actin polymerization. And this is the specific control, in which without the promoter you don't see any even bacteria attached to the cells in this particular case. So this summarizes, I don't know what happened with this, like not so intense. So yeah, no, now you can see better, huh? Yeah, I don't know what, it was like tired of projecting things. So well, you can see the image here. So this is a summary of what we have now, but we are working on systems that may allow us to induce the system by not IPTG, but other inducers that can be used in vivo. But essentially, the strain, it is in a way in which you can assemble these specific injectisomes on the surface, and then you can produce the protein of interest in a different position, and then this protein will be injected into the mammalian cell. And so what could be the application of this, especially if we combine it with the targeting that I mentioned previously? So we could deliver proteins specifically to specific cells. And not only antibody fragments, we can also think on natural effectors found in these pathogens. This pathogen has like a catalog of proteins that trigger different pathways. So you can choose something that acts on for cell killing or for inflammation, for instance, and so you can target particular pathways using this delivery of protein delivery in the cytosol of bacteria. And of course, you can introduce other type of things like enzyme, peptides for immunizations, or why not transcription factor. Has been shown that certain transcription factor can be translocated through this system and can modify the genome also of the cell. So I think there's an opportunity to do a real application with this type of bacterial engineering. So I would like to, although you have been seeing the people that has been involved in my presentation, I would like to thank especially Carlos for his work in synthetic adhesins. Valencio has developed this system also for selection, so we can select against different targets the adhesins and the nanobodies that are of our interest using directly the bacterial display system. And David was mainly responsible of the engineering of the type 3 in K-12. And I would like also to thank a friend, Professor Gerd Frankel from Imperial College, who is an expert in IPEC, EHEC, etc., and who introduced me to all this world initially, and to Victor, of course, who has been my, let's say, mentor in the past and who is still inspiring many ideas in our group. And so, thank you for your attention. And